Welcome to the video for chapter 4.5. So chapter 4.5 is all about cell membranes and ways that we move things across the membrane. So what we call cell transport. So we're going to start talking about membranes and why they're important, and then go on to how we move things across. Okay, but before we start into that, let's start with a review question. So review question number 1, which of the following are true about enzymes? They lower the activation energy of a reaction. B, they are consumed during the reaction. C, they are not consumed during the reaction. D, A and B are true. Or E, A and C are true. Okay, so pause the video and take a minute and try to find the right answer. Okay, the answer to this question is E, A and C. Okay, so enzymes, they help lower the activation energy of the reaction. That makes it speed up, it goes faster. But the really important thing to remember about enzymes, like all catalysts, okay, catalysts will speed up a reaction, but actually won't be used in the reaction, so they are not consumed. Okay, so the answers are A and C. Okay, so 4.5, our section objectives are we're going to be able to describe the structure of cellular membranes. We're going to be able to explain the concept of selective permeability when we talk about membranes. We're going to talk about how membranes compartmentalize or break down the cell into different parts and control the movement of molecules in the cell. And then we're going to be able to describe osmosis and the effect that they have on membrane compartments. Okay, so let's talk about the membrane. So the cell membrane, the plasma membrane, the cytoplasmic membrane, these are all words that mean the same thing. Okay, the cell membrane is going to act as the boundary for the cell. Okay, so it's going to create two distinct environments, the internal intracellular environment and the extracellular environment, so the environment outside of the cell. Okay, so the inside is going to be different than the outside. And this is actually really important when we talk about cells because this membrane, because it creates a boundary, is going to keep things that are supposed to be inside the cell, and keep things that are supposed to be out of the cell, outside of the cell. Okay, now we're going to go into a lot of detail about membranes in this class. Okay, we talk about membranes a lot, especially in microbiology. Okay, not only do they define the cell, any rupture or disruption to the membrane is bad news bears for the cell. Okay, if you get a hole in your membrane, that's bad. Okay, stuff in your cell, the inside of the cell is going to start to leak out. Things that should be outside are going to get in, and it spells cell death for your cell. Okay, so we're going to talk about reasons why the membrane is important. We're going to put the microbiology spin on this. We're going to talk about why they're medically important. So one of the things that we're going to talk about is how we have certain treatments that target cell membranes in things like bacteria. Okay, and just a reminder, so we create two fields. So if we look here, we have the extracellular fluid, so the area outside the cell, and we have the inside of the cell. So the fluid inside of the cell is called the cytoplasm. It's kind of a gel-like water-based fluid. It's kind of squishy. We'll talk more about the cytoplasm, I think, in chapter 5 versus here. Okay, so let's talk about reasons why we care about membranes, especially for people who are thinking about going into the medical field. Okay, so reason number 1, we have developed drugs that target these membranes. Okay, so we have some antibiotic drugs that their whole job is to disrupt the membrane. So if you've ever used triple antibiotic cream, Neosporin, bacitracin, they're all the same. Okay, the key ingredient in these is a molecule called polymyxin B. Okay, what polymyxin B does is it causes the membrane to degrade in the bacterial cell, aka it punches a hole in the membrane, and this is going to cause the cell to leak and shrivel up and die. Now, we do have some strains and species of bacteria that have evolved and they are now resistant to polymyxin B, but we'll talk about those in detail in another chapter. So if we come here, we have this pretty great graphic from the book. So we see our cartoon E. coli. Okay, we add polymyxin B, so we add our neosporin to our infected cut. Okay, what's going to happen is it's going to degrade the membrane, so it's going to punch that hole in it. So we start to see these holes form, and we start to see it shriveling up. Okay, so things are leaving it, it's going to die at some point. So what happens is polymyxin B is electrostatically attracted. Okay, so I believe it's positively charged and your cell is negatively charged. Okay, so it's going to be attracted to your membrane. Dun dun. Here we go. Okay, and what's going to happen is this charge is going to allow it to slip into the membrane, and this is how it's going to punch a hole in it, and the cell can't come back from this. Okay, so cell membranes have a lot of functions. We talked about quite a few of them in Biology 201. So other than creating a boundary, one of the things our cell membranes do is they allow the cell to communicate with other cells. Okay, so what they have on the outside is these receptors, and these receptors send molecules back and forth between cells in the nearby area, and this is how cells can talk to each other. Okay, so they use these proteins in the membrane to communicate. Another thing that happens is some of these membranes that we have on the surface of our cells, and so they're embedded in the membrane, they are going to act as identification for the cell. So like if you are an immune cell, so say a white blood cell, what you're going to do is you're going to look at the cells around you, and you're going to look for proteins that say, hey, I'm supposed to be here. We say it helps identify things that are supposed to be there as self, and invaders like bacteria and viruses as non-self. Okay, so these protein receptors in the membrane are really important in this whole function. Now we have this video. We'll watch this again later, I think in this lecture, it might be in the next one. Okay, so we have our neutrophil, our white blood cell. We have a bacteria. We have our red blood cells. So we have our white blood cell literally chasing down the bacterial cell. So the bacteria cell doesn't have the protein markers on its surface that allow the white blood cell to recognize it as being part of you. Okay, so because it doesn't have these identifying features, it's going to mark it as non-self, and it's going to seek and destroy, as we can see here, until it engulfs it and gets rid of it. Okay, cell surface markers also help in tissue development and organ development. Okay, but this is part of that cell communication and letting them work with each other. Okay, but also these receptors also create the opposite problem. Okay, they also allow a place for certain pathogens like viruses. They create a way that they can hijack your cell and use it as a host. Okay, even though we can also use them to identify pathogens as not belonging. Okay, they can use our own receptors against us. Okay, so reason number 2, we should care about membranes. Okay, membrane proteins are important for signaling, so talking, communicating, and all of that. But as I said in the previous slide, these receptors can be then used by viruses like HIV to trick the cell into letting us in. Okay, so we're going to talk about HIV. Remember, HIV is a pretty insidious disease. Okay, so this is our cell. Okay, it's going to be a T cell. It's one of our immune cells. And this is our HIV virus. Nope, get my mouse to work here. Okay, so what happens is on the outside of your T cells have a very specific protein receptor that is called CD4. Okay, so there's a protein on the outside of the HIV virus that is similar in shape to the other receptor on another T cell that is going to bind to that CD4 receptor. So what happens is this HIV cell comes and binds to this CD4 cell, and it's going to signal to the cell that it needs to bring the virus in. So the virus is going to come in, it's going to fuse with the membrane. This is going to dump the proteins and the DNA that are inside this HIV virus, and that's going to be dumped into the cytoplasm. Remember, HIV is a retrovirus. Okay, it's specifically an RNA retrovirus, so it uses something called reverse transcription. Okay, so when we dump the interior of the virus into the cell cytoplasm, we have just put RNA in there, along with reverse transcriptase, which is the enzyme that's going to go and take that RNA and convert it to DNA. Okay, so we have our reverse transcription happen. We now have DNA. DNA doesn't like to be in the cytoplasm, so it's going to move across the nuclear membrane, and it's going to go where we store our normal genome. Okay, so one of the terrible things about HIV is that it's going to not only go into your nuclear membrane, it's going to integrate itself into your DNA. Okay, so what happens now is now because it's part of your DNA, every time your DNA copies itself, it's going to copy the viral DNA as well that came from reverse transcribing the RNA from the virus. So what this is going to do is this is going to create more viral RNA. It's going to create proteins, and it's going to start to assemble those proteins and that genome together, and it's going to make new little virus particles that are going to then bud off and go to infect more healthy T cells. This is why we care about membranes and how they're built and their function, because this is actually really important in knowing how certain pathogens cause disease. So disease pathogenesis. Okay, there's a lot of viruses like the flu virus, diphtheria, that use similar mechanisms to infect a host cell. Okay, so let's talk about the structure of membranes. This should be reviewed from biology. So all cells, whether they be prokaryotic or eukaryotic, are going to have similar membrane construction. Okay, the membrane is mostly lipids and proteins. There are some carbohydrates in there that are usually attached to, say, proteins. But let's talk specifically about the lipids and proteins. Okay, the main component of the cell membrane is lipids, specifically a lipid class called the phospholipids. Okay, phospholipids are really interesting because they have a distinct shape. Okay, so we call phospholipids amphipathic, okay, meaning that they can do two things. Okay, amphi means both. And when we talk about phospholipids and them being amphipathic, what this means is that these phospholipids have both a polar and non-polar part of the molecule. Okay, so we can look here at our little diagram. So we have the hydrophilic head. This means that the head of the phospholipid is going to interact with a water environment. This can be outside the cell, this can be inside the cell. So this phospholipid hydrophilic head contains a phosphate group, a PO4, and glycerol. Okay, so this is the polar part of the molecule. And then it has a second region, which we call the hydrophobic tails. This is going to be nonpolar. Okay, so because it's hydrophobic, it doesn't want to interact with water, so it's going to do all it can to avoid it. Now, our cell membranes are actually a phospholipid bilayer, so we're going to have two phospholipid layers that make up our cell. Okay, and this is actually really important because it's going to allow those hydrophobic tails to stay away from a watery environment. So what's going to happen is the heads are going to be on the outside with the tails pointing on the inside. And what this does is this is going to create this bilayer, and it's going to allow these hydrophobic tails to avoid water. But it's also going to make our membranes selectively permeable because it's going to make it so only certain things can move across the membrane because of this hydrophobic region. Now, a lot of our phospholipids have a kink in them, like we can see here. This kink is important. This allows the lipid bilayer not to get too rigid. Okay, it keeps it fluid. Okay, membranes have different fluidity depending on a couple different factors. Some membranes usually have a consistency of salad oil, so they're not completely unviscous, but they're not like solid either. Okay, so besides our phospholipids with the kinks in them, our membranes are going to contain another lipid called cholesterol. Cholesterol is important. It's going to stabilize and minimize the effect of temperature on our cell membranes because these are lipids. Too much heat is going to cause our phospholipid bilayer to become too fluid and it's going to break apart. And if it's too cold, what's going to happen is it's going to get to be too rigid, not flexible, and it might break it and then destroy the cell as well. What cholesterol does is it allows the membrane to stay fluid without getting too rigid if it's cold and not getting, and it helps hold it together when things get too warm. Okay, so without cholesterol, our membranes aren't going to be able to hold their shape. Okay, so if we didn't have kinks in our phospholipid, okay, so if we have kinks, they don't pack too tightly, they're fluid, but if they're saturated, remember no double bonds in the hydrocarbon tails, this is going to allow the phospholipids to pack very closely together. So we have kind of a mixture between the two. So we try to keep our membrane fluid. Now, when we're using cholesterol, so at low temperatures, so to keep it from getting too rigid, the cholesterol is going to be inserted in between the phospholipids, and this is going to space them out enough that they don't pack too tightly together, and they're going to keep the membrane fluid. Okay, and when it's too hot, this is going to add stability and make it so it doesn't become too fluid. So it either keeps it from becoming too solid and packing closely together, or prevents it from falling apart once it gets too hot. Okay, cholesterol allows different organisms to live in a larger range of temperatures because it can help our membranes stay fluid. Now, if you can compare the membranes of animals here in Idaho, we are going to have a different cholesterol content than, say, an animal that lives in Antarctica. Antarctica is pretty cold. We're pretty mild in comparison. So the animal in Antarctica is going to have a higher cholesterol level in their membrane to keep it more fluid than, say, us or an animal that lives here in Idaho. Okay, so the next thing we're going to talk about is ways that we move things across the membrane. Now, we're going to talk about a couple different ways of transport. We're going to talk about mostly passive transport here, with a little bit of active transport, if you remember that from biology, but we're also going to talk about transport in 4.6. Now, the easiest way to move something across the membrane is to literally have it directly diffuse across the membrane. Okay, so diffusion is a very important definition. I would know this for this next exam. Okay, diffusion is reliant on those concentration gradients that we have talked about previously in class. So if we have a difference in concentration across the membrane, it's going to allow things to move from an area of high concentration to an area of low concentration. So we describe the concentration of something in a fluid, in a liquid or a solvent. So our solvent is going to be water in living things. So the concentration of dissolved things in the body is also measured by a unit we call molarity. So this is a chemistry unit. We're not really going to go into it because you don't need to know the math for this class. Okay, but it's going to tell us the concentration of a solute in a certain place. Okay, so we're looking for these concentration gradients. So there's difference in concentration. Okay, diffusion itself is defined as when a substance tends to move from an area of high concentration to an area of low concentration until the concentration of that solute becomes equal throughout a space. Okay, so if we look at our little animation, or not animation, our graphic here, okay, we have a concentration difference. Okay, we have a gradient. We have all these solute molecules outside. And just remember, solute is things that are dissolved in water or dissolved in any liquid. Okay, so we have all the solute out here outside of the cell, but we have none here. Okay, so what this is going to do is we're going to start to see the solute here start to move directly across or to diffuse across the membrane. So it's going to do that, and we're going to start to see some in the cytoplasm. And as time goes on, we are going to end up with the same amount of solute on both sides of the membrane, and this is where it becomes equal or hits equilibrium. Okay. Nature is always constantly trying to balance our concentration of dissolved molecules. Okay, so each different solute, each different molecule that's dissolved, so we can look at chlorine anions, just like we did in the cholera toxin example at the first of this chapter. We can look at sugar, we can look at cholesterol, we can look at a bunch of different things that are dissolved. Okay, they're each going to have their own concentration gradient. Okay, but not everything can move by diffusion across the membrane. Okay, because we have that hydrophobic non-polar region in the middle of our bilayer. Because of this, only small non-polar molecules and non-polar lipid hormones can directly diffuse through the membrane. Now we'll come back in a few slides. There's a pretty great graphic that is going to describe what can move across and what can't. So we'll come back to this. But the important part to remember for diffusion, we need small and we need nonpolar. So things like oxygen gas, CO2, and other lipid hormones, these can directly diffuse across the membrane. Okay, our lipid bilayer is going to make our plasma membrane, our cell membrane, selectively permeable. And what this means is that only certain things can cross the membrane and other things can't. Okay, size is a factor, polarity is a factor. Okay, so if we look here at this graphic, we have two solutes dissolved, and so we have two concentration gradients. Okay, we have this large blue guy, and we have this small red guy. Okay, so this small guy, let's say he's nonpolar. Okay, he's going to easily move across the membrane. Now, because this blue guy is larger than the red guy, he's going to be stopped by the membrane and he can't move through. This is what it means to be semi-permeable or selectively permeable. So some things can cross while other things can't. Okay, for the things that can't directly move by diffusion across the membrane, we have specialized structures, we have specialized proteins, and those are going to help move things like large molecules or polar molecules across our membrane, but those things that can't directly diffuse. Okay, so we're going to need specialized structures, and for some things, we're going to need energy in order to move them across the membrane. So our main component that's going to move these things that can't directly diffuse across are going to be transmembrane proteins. Okay, so this is that graphic I was talking about a few slides back. This is going to show what can selectively cross our membrane. So what can cross and what can't. Okay, so small hydrophobic, aka nonpolar molecules like oxygen gas, nitrogen gas, CO2, these easily cross the membrane by diffusion. We call them freely permeable. So they're going to go back and forth both ways. Okay, we have small uncharged, so no ions, polar molecules can kind of cross. So things like water and glycerol, they can cross the membrane. It's going to take them a while, and we're not going to get large quantities. There's actually more efficient ways to move water and glycerol across the membrane than through direct diffusion. Okay, large uncharged polar molecules, things like glucose and sucrose, they cannot cross the membrane. They're what we call impermeable. Okay, and then finally, the most polar substances in our body, things like ions, so H+, sodium+, chlorine 1-, calcium 2+, potassium 1+, these are all charged, which means they're polar. They cannot cross the membrane without help. In fact, they will never cross the membrane because they're so polar. They cannot interact with this nonpolar region, this hydrophobic region in the center of our bilayer. Okay, as I said, small nonpolar things can pass freely, large charged and polar molecules cannot. So a lot of the drugs that we use for treatment are what we call membrane permeant. Okay, so what happens when we want a drug to enter a cell, we're going to attach a weak acid or a weak base to these drugs. And what that's going to do is it's going to disrupt the membrane and it's going to allow our drugs to pass into the cell. Okay, so they can directly cross the membrane and not be stuck outside the cell. Okay, there are some drugs that are nonpolar and small enough that they can cross on their own, but a lot of them need this membrane permeant part. Okay, they need this weakly acidic or weakly basic part to help them get across. Now, things diffuse across the membrane at different rates, at different speeds. Okay, we can manipulate things to allow diffusion rates to increase or to speed up. So we have 8 factors that affect diffusion rates. Okay, you should know some of these for the next exam. Okay, the first one is our concentration gradient. So the whole reason why things can move across the membrane. The greater the difference in concentration, so if we have a really high concentration of our solute outside and very little inside, the greater the difference in those concentration gradients is going to allow the diffusion of those molecules to occur more quickly than if there was a very small difference. Okay, the size or mass of the molecule is also really important. Smaller molecules are going to diffuse more quickly than larger molecules. Okay, so even the difference between O2 and CO2, it's just one carbon, one 12.011 AMU atom. CO2 is going to diffuse more slowly than oxygen gas because it is larger. Okay, so size is a factor. So temperature is a factor as well. Diffusion happens more quickly at higher temperatures. There is a video that is linked on, I about called it Classroom, on Canvas. It's a YouTube video about diffusion. So you watch, at first you watch the diffusion of food coloring in a petri dish of water, so you can see how that starts to diffuse from an area of high concentration to low. But it also has a great portion at the end of it that shows you what happens with diffusion if you have different temperatures. So it shows you the diffusion of food coloring at a warm temperature and at a cooler temperature, and you can really see the difference in this. So I would highly recommend watching that. Okay, the next one is solvent density. So our main solvent in living systems, so like us, is water. That's what most things in our body are dissolved in. So if you're dehydrated, what this is going to do is it's going to make it really hard for things to diffuse across the membrane. So dehydration is going to increase the density of the cytoplasm, which is going to slow diffusion rates down. Okay, so the more solvent you have, aka the more water, the faster diffusion can happen across the membrane. Okay, the next one is solubility. Whether we're polar or nonpolar, as I said, small nonpolar molecules are going to diffuse faster than small polar molecules. Okay, nonpolar molecules are always going to diffuse faster. That's what solubility means. Okay, surface area. The more surface area a cell has, the more things it can bring across and the faster that things can diffuse across. Membrane thickness and distance traveled. This makes sense. It takes longer to drive across Texas than Connecticut. Okay, so if we have a thicker membrane, and different bacteria cells have different membrane thicknesses, the thicker the membrane, the slower the diffusion rate. Okay, because the greater the distance, the longer it's going to have to travel to get there. Okay. If you guys remember from biology, we talk a lot about the surface area to volume problem, because there comes a point when cells are growing that their volume is going to get too big and their surface area can't support transportation of things across the membrane. So we have to really think about our membrane thickness and our surface area when we're thinking about diffusion. And then finally, an outside influence we can use is pressure. Pressure can increase our rate of diffusion across the membrane. So this is actually how our kidney cells function. Okay, we increase the pressure and it's going to literally push things through the membrane. This is how our kidneys filter things out, and it does it really quickly. Okay, so increase the pressure always increases our diffusion rate. Okay, so let's talk about the things that can't directly pass through the membrane. they're going to need a little bit of help, and that help is going to come by the way of what we call a transmembrane protein or a channel protein. They're one and the same. Okay, so these are going to be proteins that cross both layers of our lipid bilayer. Okay, so it's going to go all the way through from the outside to the inside. Okay, they're going to make hydrophilic, so water-loving tunnels, and this is going to allow things that are polar or are charged to cross through the membrane with no problem. So things that couldn't previously cross can use these channel proteins to do so. Now, these are proteins, remember, shape denotes function. So the different channel proteins are very specific for one or just a few closely related molecules. Okay, so it only is going to bring one thing across. We have channels called aquaporins that are specific to move water back and forth across the membrane. Okay, so they're hydrophilic, which means the water, so they're polar inside, so that's going to allow the water to move back and forth quicker than it can diffuse across the membrane. Okay, some of these channel proteins are open all the time. They allow things to go back and forth. Others are gated, which means that we have to have something bind to either the inside part of the channel protein or the outside part that's going to cause a shape change that's going to allow the channel to open up to allow things to pass through. Okay, so these gated ones only open when a signal is received. Okay, these transmembrane proteins have a lot of other jobs as well. Okay, they can act as a transporter. So as we've talked here about their main function, they can be a cell receptor, they can be enzymes, they can be anchors. There's a lot of jobs that these transmembrane proteins do. But our main concern in this lecture is the fact that they allow things to cross the membrane that wouldn't be able to diffuse across. Okay, so that's talking about the solute, okay, the thing in the concentration gradient. So even with the transmembrane proteins, we still do need a concentration gradient. So one side is going to have a higher concentration than the other to allow things to move through the channel. Okay, we're going to talk about a special case of diffusion now. This is the diffusion of water across the membrane. We call it osmosis. Okay, this is what happens when we try to balance the concentration of that solute. So when we try to get it to equalize. Okay, so we're going to move water specifically from an area of low solute concentration to an area of high solute concentration. Okay, so another way to think about this, water also moves like regular diffusion. So it goes from an area of high water concentration to an area of low water concentration. Okay, but water is the solvent. So we usually talk about this in terms of the solute. Okay, so if we have a low solute concentration, that means we have a high water concentration. Okay, if we have a high solute concentration, that means we have a low water concentration. Okay, so water is going to move from this area of low solute concentration to an area of higher concentration. Okay, now the more solute the water contains, so the higher the concentration, the less likely it's going to be to move across the membrane or into an adjacent compartment. Okay, so what we can see here in this graphic is we have a high concentration of solute here and a low concentration here. So to equalize the concentrations, instead of if the solute can't move across the membrane, what's going to happen is the water itself is going to move to change the concentration. So when you increase the water content on this side, the concentration is going to reduce, and if you lower the water on this side, the concentration is going to increase, and at some point the concentrations will be the same, even if the molecule transfer hasn't happened, okay, because the water is going to make that concentration equal. Now, when we talk about osmosis, we have to talk about something called tonicity. Okay, so this is going to be what happens when water can cross the membrane, but dissolved solutes can't. Okay, we have three different types of tonicity. We have hypotonic, isotonic, and hypertonic. Okay, so let's go back to learning about what words mean. So hypo means low, and hyper means high. Okay, tonic is actually really interesting because it is referring to the solute concentration itself outside the cell. Okay, so hypotonic, isotonic, and hypertonic are going to refer to what's happening outside the cell. And then we're going to kind of predict what the water itself is going to do. Okay, so let's start with isotonic. This is our Goldilocks condition. So what happens here with an isotonic is that the concentration of the solute is going to be the same outside the cell as it is the inside the cell. Okay, we are still going to have water movement. We're going to still have water going out of the cell, and we still have water coming into the cell. But it's isotonic, or the Goldilocks condition, because the amount of water that's flowing outside of the cell, the same amount is coming in. So even though we have movement, we have no net movement. It's canceling each other out. Okay, so this is the condition that a lot of cells like to be in, is this isotonic. Now let's talk about the other type. So hypertonic, hyper means high, so hypertonic means high solute concentration outside of the cell. Okay, so we can see that here in our second picture. So we see the high solute concentration. What this is going to do is the water is going to move from an area of low solute concentration to high. Okay, so it's going to move out of the cell, which is going to cause the cell to shrink and kind of shrivel up and die. Okay, so the water is going to move out of the cell to equalize the concentration. Now, hypotonic means that we have a low solute concentration on the outside, which means our solute concentration on the inside of the cell is going to be greater than the outside. So the water is going to flow from the outside of the cell into the cell. What's going to happen is the opposite with hypertonic. Water's flowing into the cell, it's going to cause the cell to swell, and if we get too much water, our cell is going to burst or lyse. So lyse just means to break open. Okay, we need to know these different types of tonicity. Now, if we refer back to our cholera toxin example at the first of the chapter, so the cholera toxin gets in, it binds to the receptor, it gets brought in, it gets broken down. That one toxic piece is going to go through and attach to our chlorine pumps. And then what it's going to do is going to pump the chlorine anion, because it's going to turn those up. And the chlorine anions are going to be all pumped out into the extracellular matrix, so outside of the cell. And what this does is it's going to create a hypertonic condition on the outside of the cell. This is why dehydration and diarrhea happens when you have the cholera toxin. It's because all that water that's inside your cell is going to flow out, trying to equalize that concentration gradient. And if you don't do anything and you're just trying to drink water and you don't do anything to combat the toxin, you're going to just keep getting water out of your cells. It's not going to go in, and you're going to constantly have more and more diarrhea. Okay, that's why we have to use that ORT, the oral rehydration therapy. So that's going to use lots of salt and sugar, and it's going to increase the concentration of both of those inside the cell, which is then going to allow the water to go from the outside back into the inside and rehydrate the cell. Okay, you need to be able to explain each of these conditions and what happens. So on the next slide, we are going to watch an animation about tonicity.
Just as solutes tend to diffuse from regions of high concentration to regions of low concentration, so do water molecules. Osmosis is the diffusion of water across a selectively permeable membrane from regions of high water concentration to regions of low water concentration. The concentration of water in a region depends on the amount of dissolved solutes in the water. The water concentration is low when the concentration of dissolved solutes is high. Conversely, the concentration of water is high when the concentration of its dissolved solutes is low. A lipid bilayer is a selectively permeable membrane. Lipid bilayers allow water molecules to pass through, but block many dissolved solutes. Here the solutes are blocked from diffusing down their concentration gradient across the membrane, but the water molecules can cross. Osmosis across the cell membrane must be tightly controlled to preserve the form of the cell. A higher solute concentration outside the cell is called hyperosmotic or hypertonic. When the solution outside is hyperosmotic, osmosis causes a net loss of water from a cell. The cell shrinks and the cell solute concentration increases. A lower solute concentration outside the cell is called hypoosmotic or hypotonic. When the solution outside is hypotonic, osmosis causes a net uptake of water. The cell swells with water and the cell components are diluted. If enough water enters, the cell lyses. The cell membrane ruptures and disperses cell contents. When a cell is in an isotonic or isoosmotic solution, in which equal concentrations of dissolved solutes are inside the cell and out, the two are in osmotic balance and water will enter and exit the cell at equal rates. That is, no net movement of water occurs. Free-living cells, such as microbes that do not live in isotonic environments, have a variety of adaptations for survival. For instance, the ocean-dwelling bacterium, Vibrio cholerae, lives in a high-solute environment. These bacteria accumulate solutes known as osmolytes to ensure that they are isotonic to the external environment, thus preventing water loss. Other microbes live in hypotonic environments and possess a rigid cell wall external to the cell membrane. As water enters by osmosis and pushes against the cell wall, the wall resists the tension and pushes back with an equal but opposite force, called turgor pressure. Turgor prevents water movement and the cell does not lyse.
Okay, so let's talk about a real life example. A lot of you want to go into the health field. Now, one of the things, if you're a nurse or a doctor, one of the things you're going to do is you're going to be administering IVs and specifically fluid through IVs. Now, we cannot just give water directly through an IV because what's going to happen is if we add water to your bloodstream, let's talk about what it does to your red blood cells. So if we have a lot of water on the outside, we are going to dilute the solute concentration. So we're going to have a hypotonic scenario here. So what's going to happen is if we added water directly to your bloodstream, what's going to happen is the water is going to pass through the membrane of the red blood cell and it's going to cause the red blood cell to swell until it lyses. Okay, this is why IV fluids are going to contain saline. Okay, so saline is going to keep the conditions when you're giving an IV isotonic. So we have the same amount of water going in and the same amount of water going out, and the red blood cell's happy. Now, if we administer an IV that has too much salt in it, so it's high concentrated saline, this is actually bad news bears as well. Because what that does is similar to our cholera toxin example, and it's going to make it a hypertonic condition outside the red blood cell, which is then going to cause the water of the red blood cell to cross, okay, and then it's going to cause this red blood cell to shrivel up. Okay, our red blood cells are pretty finicky. They really like to have a nice proper balance. So if we were using water instead of saline, we would actually cause mass hemolysis, meaning the lysing of red blood cells. Now, if too much water through an IV can actually cause a mass hemolysis event, which will then cause the patient to die. Now, if it's just a little bit of extra water, it's not going to be as terrible, but we try to avoid this at all costs. Okay, we now know exactly what is happening in our cholera toxin example. Okay, we can now add words like hypertonic, and the hypertonic environment is going to draw the water outside of the cell. As just a reminder, you need to be able to explain what is happening in this cholera toxin case on the next exam. So this is a good slide to know. Okay, that is chapter 4.5. We'll see you again in chapter 4.6.